I would like to say thank you for meeting committee for giving me a chance, opportunity to talk about our experience working with microphysiological systems in our pharmaceutical companies. So this is my COI, and this is the outline of my talk. So today, first of all, I'd like to share our expectations to MPS from a standpoint of a pharmaceutical company. Then going to share our case studies for the use of MPS for disease modeling. Then I'm gonna touch upon future perspectives. So I'm going to start my talk by sharing our daily conversation in our lab in pharmaceutical companies. So we start exploration of 3D culture models from 2014. And at that moment, the word of MPS is not famous, and maybe the word of IPS is more famous. And I was often asked that what the differences between MPS and organ-on-a-chip. Though very recently, FDA shows a very clear definition of MPS on their website. And also, I was asked to show the clear benefit of MPS in our drug discovery. And we keep exploration of 3D culture models and get data and back to my colleague and my boss. And also I was asked like this way, oh, it's not a real organ. Of course, already know it's not a real organ for us, but I was asked like this way. And also someone like to say, oh, MPS looks like artificial. But I wanted to push back to him. It's art, but it's kind of a state-of-art technology. But anyway, very recently, the type of question come up to me is a little bit changing, which means that my colleague want to ask to me how other pharmaceutical companies can get along with MPS. And also someone want to know how FDA evaluate MPS. So I think this is the essence of a game-changing situation. So many people start to recognize the value of MPS. So going back to the definition of MPS, we submit a review articles in 2019. And in these articles, instead of giving a definition of MPS, but we proposed human organ models and categorized this into 3 parts. First one is just an isolated organ from human, like blood or liver or kidney. And the second one is human organs in animals, like human liver chimeric mice or simple tumor xenograft animals. And the third one is reconstituted organs. And we grouped organ-on-a-chip and MPS and 3D culture and organoids into this group. And this was very helpful for understanding the value of these human organ models for our drug discovery. And this cartoon would help how human organ models help our drug discovery. So as a pharmaceutical companies, we have seeds or candidate medicines for clinical trials. And animal models is a kind of a golden standard to test our hypothesis of the value of the seeds or candidate medicines. And human organ models would be very useful to work with animal models with a complementary manner. This means that when pharmacologists get a very nice data with animal models, then we can add some nice data with human organ models and add some values in viewpoint of the species differences. So this is the first use of human organ models or MPS. And the second use is translational research. MPS is very useful when we observed unexpected event in human, especially for investigating their mechanism by using human samples. So this is a kind of a translational research. And also the third use is more prospective use, which means that when we observe the change of biology or change of biomarker in human organ models, then keep exploration of to monitor this change in clinical setting. So this is more like a prospective use of these models. And to the end, these organ models could be replaced with animal models in near future or far future, but at any point. So under this, I brought this for help understanding why pharmaceutical companies need MPS. I prepared this table from these very nice papers telling the reason of failure of clinical trials of phase 2 and phase 3. And lack of efficacy and safety issue is top ranked. And the issue is there are limited or no improvement of this failure rate over the 5 years, despite of the very interesting new technology around the world. And I also brought these things for showing our expectations of LPS from pharmaceutical companies. I brought 2 articles written by a researcher or employees working for pharmaceutical companies. So top one is from our side. MPS should be very useful for investigating organ-to-organ interactions beyond cell-to-cell interactions. And the use of this system may not be limited to the assessment of DMPK profile or safety profile. This means we expected to use this for disease modeling. And another one is coming from AstraZeneca company, and they showed some issues of the unexplained toxicity in human and showed very similar expectations to MPS. So before moving to the disease modeling, I'd like to share our experience to work with MPS or organ models for toxicology field. Because this data was very impactful for me for getting more confidence to the human organ models. And I was very happy to share this experience to FDA and IQ Consortium held in their workshop on 2019. It was the timing of the soon before the outbreak, first impact of the COVID-19 situation. But this graph shows the concentration of YMXXX in various experimental settings and also clinical settings. And one thing to add, this YM3X was our candidate drugs and get entered clinical trials. However, because of hepatotoxicity, we have to terminate this project. And let's think about the situation of drug discovery or development. Before clinical trial, we dose this YM3X to rat, dog, and monkey to evaluate the toxic potential and dose to the high concentration, and we don't see the sign of hepatotoxicity. So we succeeded to enter clinical trial from the very low concentration compared with the animal settings. However, unfortunately, we started to see the elevation of ALT levels in plasma, unfortunately, and have to terminate this project. But right panel shows the result of treatment of this YM3X to human bioprinted liver models. And then we succeeded to detect the elevation of ALT enzymes from culture media. So, and this was obvious that animal models doesn't work very well to evaluate the hepatotoxic potential, but human organ models can be helpful for investigating this. And by taking this data, I imagined the success future of the MPS in near future. So then we moved to the disease modeling things with MPS. So this slide shows a very simple concept of disease modeling. The first thing to do is attempt to reconstitute of human organs by elements of MPS, like a different type of cell or nice device, or physical cues, like shear stress or stretch or something. Then if we can capture organ function or part of organ biology, then we want to further manipulate this organ for giving some damage by adding some biological stimulus or pathogen, or in some case, it's gene editing or disease cell type. And then if we can observe some differences between this healthy organ and damaged organ, then we can name it as a disease modeling. And then we can test our test article to see the recovery or prevention effect of our candidate drugs. This is a concept of disease modeling. And in other case, we sometimes to see the very nice biology during the development of these organ models, like angiogenesis or T-cell migration. And if we observe these things, we want to test to work with our candidate drugs to see how these candidate drugs can change our biology or their biology. So this is the concept of mode of action research. And I brought the first case of angiogenesis. Yeah, I won't go detail about this because Paul already mentioned, but the left cartoon shows the biology of angiogenesis. It is believed to be started from a gradient of angiogenic factors. Then blood vessel can respond to this gradient, then start to sprout. And these angiogenic factors sometimes coming from tumor factor, tumor component. And this device is very nice to capture this biology because of the gel things in this middle lane. So first, we put some gels in this middle lane, and then add angiogenic factors in the bottom lane. Then we can make a gradient of these angiogenic factors because of the high viscous ECM. Then the Hubeck cells on the top lane can respond to this gradient and then start sprouting. Then this is the actual data. We can induce this angiogenesis by sprouting cocktail added in the bottom lane. And then we want to work with some test articles. The actual data working with tyrosine kinase inhibitor was shared in this session in this Wednesday by Takama et al. And one thing to add is that we can reproduce this data on our end in addition to the Mimetas side. So, and this is another case to model renal tubular damage by ischemia reperfusion using MPS. So in this research, we take this culture setting, which means Hubeck in top lane and renal epithelial RPTEC in the bottom lane. And after characterization of this model by immunohistochemistry, we tried to capture ischemia and reperfusion. And for mimicking ischemia conditions, we put various type of culture condition. For example, we put this in the low oxygen incubator and removed glucose from culture media. And we put this on the static condition to give no fluidic flow. Then I'm going to share the result. So I'm going to share the result of this ischemia reperfusion, but I have to add something. So in this experiment, RPTEC kidney epithelial is a top lane, and Hubeck is a bottom lane. And the left side is a control situation, and we tested the effect of adenosine to see the effect of their effect from the renal damage. And after 24 hours, we put this plate on the ischemic condition and then return to the normal culture condition. Then first 24 hours ischemic condition, there are no obvious differences due to condition. However, in the reperfusion stage, we see a detachment of cells in controlled situation and increasing this green color, which is a representation of an increase of Caspar-7 activity. This means that renal RPTEC was damaged by the stimulus of ischemia and reperfusion. And more interestingly, adenosine have a prevention effect to this ischemia and reperfusion because of the very nice morphology and very limited green things. So this is a case of our disease modeling and compound testing. And I brought one interesting paper because this should be very useful for accelerating our drug discovery, because this table shows the very active users of MPS from pharmaceutical companies and also their providers. And one good thing of this table is context of use of MPS is well summarized. So this type of exposure will help acceleration of the use of MPS in our drug discovery. So I move to the final part for the future perspectives for increase the use of MPS in drug discovery and development more and more. One point is automation of MPS preparation and assay. As you know, one of the issues of the MPS is their robustness and reproducibility of the assay and readout. So we introduced a double arm robot into our lab and work with 3D culture models. And I'm going to start this movie, but I won't go detail, just feel the movement of this robot. And bottom panel is the actual data. When we conduct some manual study, we can induce a collagen 1A1 messenger RNA after treatment of TGF-beta incubation. This is a simple case of a disease modeling of fibrosis. But we can get this data, but when we look at the cellular morphology in well by well, some well shows a kind of a broken spheroid structure. So we trained this machine, this robot, to the especially gentle pipetting and gentle sample collection. And as a result, we able to see a very nice morphology of spheroid and also get a biological response. And this machine is combined with very nice CO2 incubator, so they don't see the big edge effect. So we can work with a hollow 96 well plate of this spheroid type spheroid generator. And another important point is to use MPS data for regulatory submission. And this topic was very well discussed in other session, so I don't go detail, but I would like to congratulate Sanofi company and the provider of this very fantastic MPS device. And this should help patients suffering from CIDP in this case. And we would like to follow this type of research activity in our end. And this is a take-home message that is a very simple. MPS is already being implemented in pharmaceutical companies. And for supporting these messages, I analyzed the landscape of MPS in Astras, starting from 2014. So there are 17 ongoing projects working with human organ models at this moment. And the first contribution was ADME part, and we also contributed with the safety part. But majority of the work with the efficacy study and also MOA study. And we work with small molecules and antibody, but not limited to those, but we work with other new type of biologics or modalities. And our major platform is Mimetas, but not limited to Mimetas, because we work with project for fit for purpose manner. So in some case, we take academic technology that is before commercialization. So that's all of my presentation, and I would like to appreciate a lot of collaborators and business partners, and also founders and consortium. And we cannot do this on our end, so we need this type of help, and say thank you for this, all friends. So that's all my presentation, and thank you for your attention.